All right, we'll begin. First, I'd just like to do the Acknowledgement of Country. So I'd like to begin by acknowledging the traditional owners and custodians of the land in which we meet today, the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present, and from other communities who may be here today. So thank you for joining us. Thank you for joining the people online on Slido. It's my great pleasure to introduce Daryl to give his completion seminar. Working with Daryl has been an absolute amazing pleasure. I first met Daryl when we engaged a company that he worked for, DS, to do a small project, and it was actually between proteomics and imaging. I think Lockie, I hope you're online. This is going back a number of years now, and Daryl was the CTO at DS. So we got to meet him in quite a leadership role, where he was helping oversee one of the teams that we were working with. And he started becoming more actively involved, and then through more and more conversations, Daryl actually reached out and said, you know, is there a potential to do a PhD? And I was a little bit taken aback, knowing Daryl in the role that he was in, making that transition into being a student after being in such a senior role, I think is quite commendable. And Daryl we were just discussing now, that by doing something like taking on a PhD, you're really putting yourself out there. You are challenging yourself, you're making yourself, you are really, you're not making life easier for yourself. I think Daryl has had not the easiest PhD. I think we started out with some amazing big grand visions, and I think where we ended up probably wasn't quite where we thought we were going to get to, but where he has ended up, I think is an amazing place. I think that the stuff that he's done is going to be an amazing contribution to the field. I think it's already being picked up by the labs in the community. Brooker, so one of the companies that run the instrument that Daryl's been building software for, have been incredibly impressed with the work that he's done. To talk a little bit about Daryl, so through his education, he started out as an engineer in electronic engineering. He then did a master's at Monash, focusing on computer systems, neural nets, and speech recognition. And this, when it wasn't sort of famous and the thing to do, this was back in 1994 when he submitted, so well ahead of his time. And today, Daryl's research project is really focusing on developing novel approaches, so making the most of these new instruments that we have access to and trying to make more sense of the data that's there. So without further ado, we'll get Daryl up here talking, and hopefully there'll be plenty of time for questions after. So please join me in welcoming Daryl.
Thanks for the introduction, Andrew. I used to joke with Andrew that I wanted to do a PhD because my life wasn't complicated enough, and so I wanted to fix that, and here we are. So thank you for taking the time to come and listen to my talk, and the people online as well. So the field of proteomics is the study of an organism's proteome, the collection of proteins that are found within it. And every system and organ in the human body is enabled by proteins. They form the immune system, they help us breathe and move, they help us digest food, they form our structure. So studying the proteome provides an opportunity to have a window into our state of health. And the proteome is not static, it's a constant state of flux. External factors constantly influence the proteome either directly or via the genome. Influencing factors include diet, stress, age, level of fitness. These factors can affect the relative amounts of proteins in our body fluids and tissues. So the proteome also provides a view of how our health changes over time. This is a chart of the abundance of proteins found in blood plasma. The x-axis is the proteins detected. Their abundance is on the y-axis, and it's a log scale. So you can see that it spans 12 orders of magnitude from the most abundant protein to the least abundant. The proteins commonly used for diagnostics, which is on the left-hand side of the figure, are easier to detect because they're much more abundant. The less abundant proteins on the right may well be more diagnostically useful, especially in combination with other proteins, but they are much harder to consistently detect. And one of the challenges in plasma proteomics and the development of novel biomarkers is the limited dynamic range of instruments. Proteins are studied with a mass spectrometer in a workflow like this. First, proteins are broken down into peptides with an enzyme so that we can study them in the instrument. In mass spectrometry, the goal is to separate molecules as much as possible. And the first step of separation is done with liquid chromatography. In liquid chromatography, the sample is pumped into a column. Peptides are separated in the column according to their affinity with water. Some take longer than others, depending on the level of interaction along the way. The time it takes for a peptide to travel through the column is called its retention time, and the movement of a sample through the column completely is called the elution. Following on from liquid chromatography, the peptides are ionized so they can be manipulated in the instrument. In electrospray ionization, the analyte is converted from the liquid phase to the gas phase through evaporation, in which the molecules acquire a charge and become ions. In this process, a peptide molecule may acquire one or more excess protons. The number of protons they acquire defines their charge state, so a charge 2 peptide means it acquired 2 protons during ionization. Following ionization, the ions are pushed into a mass filter called a quadrupole. And a quadrupole is a set of 4 electrodes that can control the path of ions by manipulating the voltage on those electrodes. Very precise ion mass ranges can be selected for transmission through to the end. The ions not selected and filtered out just fly off into the vacuum. After the quadrupole, the ions then move into the collision cell. The ions at this point are called precursor ions. In the collision cell, precursor ions are smashed apart by colliding them with gas molecules, creating fragment ions. And this is done so that we can analyse their composition. The ions then move onto the flight tube where they are pulsed upwards and reflected back down to the detector. The number of ions hitting the detector is a measure of their intensity or their abundance. The time of flight is proportional to their mass and their charge. And for each ion, the instrument produces a value of the ratio mass over charge and its intensity. At the end of this process, the instrument produces data that provides the mass spectra of the precursors and their fragments. A mass spectra is a plot of that MZ value, mass over charge and intensity. And those spectra are used to identify the peptides. To do that, the attributes of the ions must be determined. For each precursor ion, the mass to charge ratio, or M over Z, the retention time, and the intensity are determined. And likewise for the fragment ions. The precursor and fragment ions are taken together then and matched against a peptide library. Peptides are identified on the basis of the closeness of the match. So rather than dealing in absolutes, we are really dealing with levels of confidence. Proteins are inferred from the peptides that we can identify. Just as peptide identification is a level of confidence, so is protein inference. Peptides may belong to one or more proteins, so it's a process of finding the most likely combination of peptides. And the main thing to take from this is that identifying more peptides more confidently leads to more confident protein identification. This is a figure from a study of peptide detection versus identification. Although instruments can detect many peptides, many of those are not targeted for fragmentation. And of those targeted, a bit more than half are identifiable. So overall, only 10% of peptide features detected were identified, and they are mostly at the upper levels of abundance. This is a problem because it limits our ability to delve deeply into the proteome. And there are two main underlying causes of this problem. The first is evident in this visualization. This is some raw data from the instrument as a complex sample elutes from the chromatography. It's a small section of the data, but there are many peptides overlapping. So the challenge is to unambiguously tease the spectra apart to confidently identify each one. The second underlying cause of the problem is related to how the instrument acquires data. During acquisition, the instrument selects precursors for fragmentation, and it only has a few milliseconds to find and select them. And it does this on the basis of their intensity. The fragment ions are an important part of identification, as we saw previously, so only choosing more abundant peptides leads to a bias. So there are a few points that I want to highlight before we go further. The first one is that we need to go deeper into the proteome to discover novel biomarkers. The second is that identifying more peptides more confidently is a critical part of that. And then thirdly, despite a lot of investment over the last decade, the pace of novel biomarker discovery has been hampered by our ability to obtain more depth from analysis platforms. So in my PhD project, I set out with two main aims. The first, to take advantage of the special features of the Timstoff, a new instrument, and I'll explain what makes it special in a moment. And my second aim was to contribute this work as open source software, and I'll explain why that's important in a moment as well. In 2016, Brooker released the Timstoff. One of the reasons it's special is that in addition to measuring ion mass-to-charge ratio, it also measures the three-dimensional shape of ions, called their collisional cross-section, or CCS. And this is useful because in a very complex sample, it's difficult to resolve peptides from their mass-to-charge ratio alone. In the Timstoff, the ions are injected into an ion mobility cell to separate them according to their collisional cross-section. A gas pushes the ions forward with a force proportional to their shape. So just as a sailing boat with a bigger sail gets pushed along faster, so it is with the larger cross-section. And then an electrical field opposes them based on their charge. And by manipulating that field, the ions are released according to their shape through to the mass filter and beyond to the flight tube, and they hit the detector. In the data from the instrument, as the elution proceeds, we can see M over Z as before, but also the extra dimension. So where peptides overlap in M over Z, they can be separated with their collisional cross-section. And this extra dimension required a different approach for processing the data. These days, there are quite a few choices for analysing Timstoff data. So you might ask yourself, why didn't we just use one of those? First of all, none of them existed in 2017 when I started on the project. And secondly, the first to come out with Timstoff support was MaxQuant, a popular program that had been around before that, but Timstoff support was new. But to make the task manageable, it discards 2/3 of the ion mobility data by default. And as Pepe famously once said, Max Quant treats ion mobility as an inconvenience rather than an asset. I'm not sure he wanted that repeated in a seminar like this, though. So we wanted to develop new algorithms that embraced the richness of the instrument, even though that was much more challenging. And without open source, a researcher must rely on a description of the algorithm in prose by reading the paper, which limits their ability to learn how others tackle the complexity of the instrument. So my second aim was to contribute open source algorithms that analyse Timstoff data and fully embrace its richness, and offer these algorithms to other researchers, especially those trying to get their head around how to consume Timstoff data. And I think this is important for a few reasons. Firstly, open source provides clarity. It gives a researcher the ability to look at the actual code and the details of the algorithm rather than trying to understand it from a paper. It removes ambiguity. The second is that all software has errors, some big, some small, and so opening the code allows more eyes to look over it and possibly find better ways of doing things. And then thirdly, by accepting code improvements and new features from the community, the original authors can make more progress more quickly. So peptide detection is a pattern recognition problem, and patterns have a predictable pattern. But what do they really look like in real data? This visualization of the raw data shows the scale of the data and the 4 dimensions. It shows the breadth of M over Z, the CCS, and a few seconds of the elution. There are 2 clouds of peptides we can see here. The first at the top are the charge 1 peptides, and toward the bottom is the peptides with charge 2 or more. And even at this level of complexity, we can see the patterns, but also the sparsity of the data. When we zoom in, we can see that there are distinct patterns. The blue rectangles are highlighting potential peptides, and there are a few things to note here. Firstly, peptides can overlap in time and mobility. The data is a sparse array of intensity readings with no inherent structure, which has always been true, but with ion mobility added in, we've traded another degree of separation for more complexity in the task of detecting features. And a peptide feature has the characteristic lines that come and go through time. Those parallel lines are due to the different isotopes of atoms in the ion. In this example, 2 amino acids have formed a bond to become a very simple peptide. And in the peptide molecule, there are 4 carbon atoms. In nature, carbon has 2 stable isotopes, carbon-12 being the most abundant, and carbon-13 also stable, and somewhat common at 1.1% of all carbon. Carbon-14 occurs in nature, but it's radioactive. The isotopes differ, of course, by an extra neutron. So for every carbon atom, such as the 4 in our simple peptide molecule, there's about a 1 in 100 chance that atom will be a carbon-13 with that extra neutron. And the instrument is sensitive enough to pick up those differences, and that's the reason we see those parallel lines in the data. They're formed by the isotopes and the mass of that extra neutron. The leftmost peak represents ions with all carbon-12 and no carbon-13, and it's called the monoisotopic peak. The rightmost peak records the ions with all carbon-13 and no carbon-12, and it's the greatest mass for the peptide because it has those extra 4 neutrons. The height of each peak is a reflection of the probability of each carbon in the peptide being carbon-13. As mz is a ratio, with the denominator being charged, the spacing between peaks is due to the charge state. And you might recall that the charge state is from the extra protons acquired during ionization. So for a charge 1 peptide, when the denominator is 1, the spacing between the peaks will equal the mass of a carbon neutron. And from this pattern, we can calculate the peptide's mass and its charge state, which are two really important attributes for its identification. For clarity, let's take an artificial example of an isolated peptide without noise or interference from nearby peptides. We've seen that a peptide appears as that series of isotopic peaks. The peaks have different intensities, decreasing with increasing m/z. They should be aligned in mobility, as we'd expect, because they're coming from the same molecule. And as we turn around into the mz dimension, we should see that they are evenly spaced in m/z. This is an example of a precursor ion, and this is exactly what we're looking for. Assisting the task of detecting peptides is the fact that they appear in a defined pattern. However, they are still challenging to find. Firstly, they overlap with each other in every dimension. Their structure becomes less defined with low abundance, and noise can look very much like a peptide. And the data that they appear in is both fast and sparse. We've established the importance of finding as many peptides as possible across a wide dynamic range, and we've seen that they have a distinct pattern. But it's a vast data landscape we need to work with. How do we do that? How do we go about finding them? There are three main methods, and in this project I developed processing software that applied each approach. First, I'll explain the concept of each method and then talk about my implementation of it. The idea of targeted detection is to reduce the search space by providing a guide. And one such guide is the record the instrument makes when it selects precursors. We can use that to constrain the search space to look for peptides. Targeted detection looks for features in that area and uses a peptide library to identify them. And once identified, they can be annotated in the raw data. In targeted extraction, on the other hand, the location of each peptide in the library is predicted, usually with machine learning models. The peptides are extracted from that location, and if their attributes match what's in the library, that peptide is considered to be identified, and it can then be annotated in the raw data. And thirdly, in de novo detection, the algorithm goes hunting for peptides in the vastness of the data space. It has no expectation of the peptides it will find or where they might be. Instead, it's looking for patterns. When it finds a peptide, it looks in a peptide library for a match of those attributes. It's much more computationally expensive, but on the upside, it's unconstrained, and so the benefit is much more thoroughness. Starting with an original idea from Giuseppe, I developed a Timstoff processing pipeline called Targeted Feature Detection and Extraction, or TFDE. As the name suggests, TFDE combines the first two approaches I mentioned, targeted detection and targeted extraction. Each analysis by the instrument is called a run, so for an experiment of technical replicates, there is a run for each analysis of each replicate. There are two halves to the pipeline. The top half shows the targeted detection process, and it's used to build a library of the peptides detected and identified in at least one run of the experiment. The bottom half uses the peptide library to train machine learning models for targeted extraction of the peptides from all the runs. So top half is about collating peptides we've seen at least once, and the bottom half is about trying to extract them from all the runs. Starting with the top half, the reduced search space is the region around instrument-selected precursors. And these regions are called precursor cuboids. Precursor cuboids are formed by extending each dimension around the instrument-selected precursor region. The idea is that within this expanded region there will be other precursors that would have been co-fragmented because they were close by. For each cuboid, the task is to resolve each precursor's peaks in each of the 3 dimensions. And we know that the peaks are Gaussian or Gaussian-like in MZ, CCS and retention time. And we need to know their apex and to separate them from nearby interfering peaks. Looking at the plane of MZ and CCS here, the blue rectangle shows the instrument selected region and the red rectangle shows the expanded region. We can see the selected precursor here, this intense point there, and there's another separate precursor here. For each precursor cuboid, looking at the MZ intensity plane, we can see peaks in that characteristic peptide pattern. The peaks are in the form of raw points at this stage, so we need each one to be resolved to a single isotopic peak, as shown in panel B, by taking an intensity-weighted centroid of M over Z. And then lastly, the raw points are now in the form of a series of isolated peaks. Those resolved isotopic peaks are passed to the feature deconvolution algorithm, which fits a theoretical peptide model and finds viable peptides in the spectra. A candidate peptide is called a feature, and we can see here it's proposing those 3 features, and it specifies the candidate monoisotopic peak and the charge state for each one. The raw points in each candidate monoisotopic peak must then be resolved in the CCS and retention time dimensions. And this is done with smoothing and curve fitting so that the apex can be determined. The monoisotopic peak is expected to be Gaussians in the MZ dimension, and this we can check with correlation. So having processed all the precursor cuboids in that way, the peptides identified from the targeted detection process are collated into a library. The next step is to try and find in all the runs each peptide that we've got in our library, and targeted extraction is used to do that. To estimate the coordinates of each peptide, machine learning models are trained from each prior peptide observation. These models take the inputs X in the table there and estimate the delta from the experiment mean Y. The prediction mean absolute error calculated from the actual feature extracted is very low, so these models are very precise. A key concept here is that unlike other approaches that manipulate and align the data, this approach preserves the data in its original form. We're creating a lens through which to look at the data, just as we wear glasses when our eyesight needs adjusting. To evaluate its performance, we mixed 3 proteomes, yeast, human and coli, and 10 technical replicates were created and processed in the instrument. Before I talk about the results, I need to talk about the missing values problem in proteomics. We used 10 technical replicates to evaluate TFDE's ability to minimise missing values. The expectation is that if a peptide is identified in one technical replicate, it will be identified in all of the others as well, as they are essentially the same sample. This doesn't happen in practice for a few reasons, such as the instrument won't always select the same precursor across runs, and there is intensity variation between runs so that peptide may not have been detected. And when that happens, it's called a missing value. Minimising the values that are missing means we can be more confident in the identification, and we have more observations from which to determine each peptide's abundance. This figure shows the number of runs in which the proportion of all peptides can be quantified. I'm comparing TFDE in the blue line, and that's the process that I wrote, with popular applications MaxQuant in orange and MS-Fragor in green. MaxQuant and MS-Fragor have dropped off by 10% in more than 3 runs, but TFDE holds on for 8 or more runs. And so we can see that TFDE has far fewer missing values compared to MaxQuant and MS-Fragor. When the peptides are sorted by intensity, if we take the lowest 500, we can see TFDE is able to detect these peptides and have fewer missing values than MaxQuant and MS-Fragor. Having fewer missing values in the lowest 500 peptides means the technique is more sensitive at lower abundance, which is what we want. So far, I've described my implementation of targeted detection and targeted extraction, which together form TFDE. Next, I'll describe 3D-ID, a de novo feature detector that I also developed as part of this project. And the name comes from 3-dimensional intensity descent, as it starts with the most intense 3D feature and works its way down. In de novo detection, we have no guidance about where the peptides are, so 3D-ID divides the whole raw data space into small cuboids called voxels. A voxel is a volumetric or a 3-dimensional pixel. And the idea is to quickly locate areas of intensity because that's where the peptides likely are. Each voxel captures a number of raw points. The raw points are summed and the voxels are sorted by decreasing summed intensity and processed in that order down to a defined threshold. In a typical run, there are hundreds of millions of these voxels. Many of them are empty though, so because the data is sparse. But 3D-ID quickly finds those that are not empty. This is an example of how a voxel is processed. The voxel is considered to be at or near the apex of an intense peak, so the peak is traced out in the CCS dimension to find the valleys on either side. In the MZ dimension, 3D-ID looks for peaks to the left and to the right, consistent with that isotopic spacing for each charge state. And now we have the extent of a peptide feature in M/Z and CCS. Panel A is showing how the base peak is segmented in CCS, and it's the same for the retention time dimension in panel B. Now that the feature is segmented in 3 dimensions, it can be presented as a simple mass spectra, as shown in panel C. The features attributes monoisotopic mass, charge, apex in retention time and CCS are now known, and they can be searched against a peptide library for identification as before. But how do we make sure the detections are truly peptides and not noise masquerading as a peptide? I constructed a neural network classifier that takes quality metrics and determines whether the detections are likely identifiable. This idea was inspired by prior work by Bin Ma's lab, but they didn't have CCS in their data, and so I added the mobility coefficient, a quality metric for how well the peaks align in the CCS dimension. The classifier was trained on candidate features from TFDE that were identified and a selection of those that were not. The output of the classifier is a score indicating whether a feature with these attributes is likely to be identified. And the model has good accuracy on the holdout test set. Even though 3DID looks for peptides everywhere, for the sake of comparison here, 3DID's features were constrained to the same search space as MaxQuant and TFDE. And there are 2 points I'd like to highlight here. Firstly, if we consider MaxQuant to be the reference, 3DID finds many of the same peptides and thus validating its accuracy. And secondly, 3DID finds more peptides than MaxQuant when it's constrained to the same search space. This is a histogram of peptides detected by abundance. The blue is all the peptides 3DID detected. The orange shows those peptides within the regions selected for fragmentation, and the same space that MaxQuant and TFDE are working inside or with, and the green shows the peptides that were identified. Not many of those selected for fragmentation were identified, possibly because they were not fragmented, or possibly because they couldn't be identified from their fragments. It's evident that 3DID is detecting many peptides in the lower end of abundance that were missed by the other approaches. And this underlines the exciting potential of this approach, as it means that there's much more proteome depth to explore. I presented the 3 ways to find precursor ions, and I've described my implementation of them. TFDE uses targeted detection to reduce the search space and targeted extraction to significantly reduce the missing values. 3DID uses de novo detection to find many more peptides of lower abundance that were otherwise missed by other approaches. Returning to my thesis aims, I've described software that I built to harness the sensitivity of the Timstoff, and that software and the data produced is now shared for other researchers to use in their own work. The code's on GitHub and the data is shared on Proteome Exchange. In closing, I'd like to offer some thoughts on potential applications of this work. In the proteomics field, targeted extraction is becoming the more popular method of analysing mass spectrometry data because of its speed and reproducibility. And we've seen the importance of the peptide library in targeted extraction. We can only extract what's in the library. But how are these libraries generated in the first place? The gold standard is currently to use fractions, where the sample is separated with chromatography and each fraction is analysed in its own separate run. This is time intensive and sample intensive, and it also has the drawback that matrix effects are altered. We need a better way of generating libraries. 3DID's comprehensiveness, combined with the ability of machine learning models to learn about sample variation, promises to be a better way. And by developing and sharing 3DID, I'm hopeful it could contribute to future research. No PhD project is an island, and I'd like to acknowledge the support of these people. Firstly, Andrew and Pepe for bringing me into the world of proteomics. Our regular Monday morning meetings after the lab meeting was certainly one of the most enjoyable parts of the PhD project, drinking decent coffee and talking about ideas, mass spectrometry, and having some laughs at the same time. And although he was distant in Adelaide, Anton introduced me to some valuable signal processing ideas, especially in the early days. My advisory committee for keeping me on the track to thesis submission, Sue and Keely in the Scientific Education Office for helping me navigate the university processes, my colleagues in the lab, especially Jared and Laura, who I think sat through every one of my lab meeting updates in the first couple of years especially, and always asked difficult questions, which certainly helped my thinking about the problem. My fellow travellers on the journey toward hybrid nirvana, Ahmed, Pedro and Saror. Liam Fernley, who oriented me in the world of scientific computing and wisely talked me out of using FPGAs. Brooker R&D, especially Sven and Michael, we were having quite regular catch-ups. They gave me a lot of technical support and a lot of encouragement as well. Joe and Clancy at Dias gave me the encouragement to start the PhD and gave me time to spend here at WeHi during the working hours. And Jaslee, my wife, our daughter Gemma and son Liam, who inspired me to start the PhD, keep going and to finish. Thank you.
Thanks, Daryl, for the wonderful overview. We have plenty of time for questions. I'll just remind people that are watching on Slido to ask your questions. I haven't seen any come through yet, but I'll open the floor for questions. Go on, Gordon. I'll bring the microphone over so people at home can hear.
So thanks, Daryl. For your open source software, what requirements would people have to run? Would have to run that? Does it assume data from a particular machine or a wide variety of machines? Does it assume a particular software setup, particular software language? Can you just delineate that a little bit?
Yeah, so it was a design goal to make it runnable on a desktop PC. We didn't have a lot of success with exploiting that parallelism of the cuboids in the cloud, but the average researcher that I had in mind, I guess I didn't want a high bar with that sort of big fat credit card. I wanted people to be able to download it, run it on an ordinary PC. So I've been running or testing it on an ordinary PC with like 12 electrical cores and 64 gig of RAM. It's not a high flying machine by any means. And the neural network is just using standard GPU, and it's a Python environment managed by Conda. So I kind of wanted to keep the level of entry to people wanting to use it as low as possible, and if they want to translate it to other languages, that would be fine. But the reason I was targeting Python was just to make it, I wasn't just targeting bioinformaticians necessarily, I was also interested in making it accessible to data scientists in the world that I came from. Probably Python is more common. So that was like the reason for that design goal.
I've had a request for you to speak into the mic when you're answering questions, Daryl.
Sorry.
That's all right. We're still waiting on questions. Please do. I can see there's 34 people still online from home. Please do submit questions. We haven't got too many people in the audience, but please. Great. Yep, I'll just bring the mic over.
I'll have to forgive the extreme ignorance since I am predominantly a biologist and I normally stay well away from this end of the mass spec. But I guess for your first part was the TFDE, and you were sort of saying that it detects more of the lower abundance peptides. That was what I was getting out of what you were saying there. I guess one of the things that I found sort of doing data analysis with sort of different softwares that as they've developed over time is once we start getting these softwares where they sort of detect these lower abundance things, that we often get what we as biologists think are errors, as in detections of knockout proteins, detections of like other issues of like male proteins in a female sample, for example. So I was wondering with your software whether you've done any tests in this kind of system to look at, like I know you sort of did the computational test for sort of how real we think this is, but have you done any biological tests for stuff that sort of, do you start throwing up false positives at the low end of the scale?
Right, yeah, so I think the chart I showed in TFDE being sensitive to lower abundance peptides was about missing values, and I guess they are identifications taking into account a false discovery rate of 10%, which is standard for peptide identification. So that's after they've passed that threshold, and it's sorting them by the lowest that have passed that threshold. So they are reasonably confident identifications, but they are really low intensity. So I think we can be, well, I think that sort of passes the bar of scrutiny in terms of false discovery. Sorry, I can't.
Sorry. It's if you think the first threshold, like the first false discovery threshold, is probably the problem I'm seeing in the other style of software. Like if it passes the first threshold, then potentially got problems either way. Is that? So I'm really not a technician on these, but as a biologist, that's what I care about when I start seeing things that we don't think are real.
Sorry, I have to admit I have hearing loss, and so I didn't catch most of that.
Sorry.
You want to try the microphone?
Put the mask on, it doesn't help. So what you're saying is that the stuff that I'm seeing is sort of failed the first threshold. So in your software, the extra detection after we've passed that first threshold is sort of, it's really dependent on the first false discovery rate. And so you think that's
the standard bar for peptide identification. We consider that to be fairly confident.
So is your threshold sort of calculated differently to any other sort of software, like for the first threshold then? Because if that's the one that matters, like I'm...
Sorry, is it different what?
Yeah, I guess my question does come down to sort of, have you tested in biological systems whether your software is doing well with false discovery? Like,
right, yeah. Well,
that's kind of the question.
It's always a challenge, especially when you're dealing with that low intensity stuff. But I think, yeah, the standard accepted bar is that 10% of false discovery. So I had another point too, and that was, I guess, related to that question was the detections of 3DID. Because I couldn't identify them, that neural network classifier was trying to classify whether they were identifiable based on identifications with the TFDE pipeline. And you might have noticed that it had an 86% accuracy, and so that's probably seeing a little bit high for or the potential for false discovery, but there are still a lot more than, that 4% above and beyond what other methods are finding. So that was the rationale there.
Okay, we do have a question that's come through from Kelly. How does the compute requirements of what you have developed compare to other software packages?
Say compared to MaxQuant. MaxQuant, I guess, was my point of reference. It runs on a commodity PC, or it can. It's happier with more cores, of course. And that was kind of my point of reference. So MaxQuant's written in C++, and I we talked a lot about that in the early days about the implementation language, and I felt, and I still feel that by writing it in Python, I'm making it more accessible. It is more probably resource intensive than Max quite a bit in terms of memory. Not so much cores. It's probably on about a par, I would say, in terms of processing time for the number of cores. But it uses a bit more memory, and that's probably the Python, I guess, price of entry a bit. And so that it goes back to Gordon's question about the design goal, I guess, that really keen for it to be able to run on the same sort of hardware as MaxQuant, because that's like the free, popular alternative.
Great. Now I've got another question from Kelly, a little bit controversial, but if you were to embark on the same project today, would you approach it differently?
Well, I guess today I put up that table of all the software choices that are available now to process Timstoff data, and that table was empty when I started. So I guess now I'd probably have the luxury of being able to focus on like a front-end feature detector and give that off to something else to finish off the the protein inference and the peptide identification, and probably could even go straight to targeted extraction and things like that. So with sort of that luxury of all those other available packages today, it probably does mean you'd start with a different approach, because there's a lot more available. But 2017 or late 2016, when I was scoping out the project, it was Brave New World.
The Wild West. All right, I think we're right out of questions. We're almost to time, but it just leaves me to thank Daryl for being a wonderful student, giving a wonderful presentation today, in particular, getting such an amazing overview. I think it is a hard thing to communicate. You're sort of very deep in a quite a challenging field, and I think you've done a wonderful job of communicating that today. I would like to just mention, before we end up with the final thank you, just to mention that this is for the first time, so people here, you should be excited. We have refreshments outside for everyone that's here in person today. If you're listening in your lab on Slido, please come up and join us in the tea room to celebrate. But thank you, Daryl, and congratulations.
Thank you.
Please join me in thanking Daryl.